Hello, and now that we have finished up and we've talked about DNA replication, transcription, and translation, let's put it all together and let's actually go from DNA to RNA to protein. So let's use the steps that we've learned individually to go through what actually takes place in your cells when we produce a protein. So we'll do a quick review, and if we look here, we see we've got the double helix of DNA. So in DNA replication, we've got complementary base pairing, and what we've got to remember is that adenine always goes with thymine, cytosine always goes with guanine. So A with T, C with G. So if we have, there we go, a strand of DNA, here's a short segment of double-stranded DNA, and we split it, which would happen during DNA replication, what we get is two single strands, and the complementary base pairs would fill in. So let's look at the upper strand first. So what would happen is we would build it from 5' to 3'. So we would start down this end, and complementary to A would be a T. T would give us an A. C would give us a G. T would give us an A. A would give us a T. Another T, a G, and a C. So there would be our complementary strand to that, and we would do the same thing on the bottom again, starting at the 5' end. Remember, they'd run anti-parallel, so complementary to a C would be a G, a C, A, T, C, T, and A. So now, if we're doing DNA replication, you should be able to take any segment, realize that they are anti-parallel, and match the complementary base pairs to each one. So there's an example of DNA replication. Now let's look at what happens of the transcription of RNA. And so as a quick review, the one thing we've got to remember is cytosine still goes with guanine, but now adenine doesn't pair with thymine, it pairs with uracil. So if we look at the example over here on the left, here is our sense strand of DNA. Here's the one that's going to be read. So G gives us a C, T gives us an A, C gives us a G, but right here, here we go, A gives us a U now. So that's going to give us our mRNA strand. So let's try an example of that. So what I did is, from this diagram, I started with our template strand, our sense strand, and I started right here with this G. So down below, we've got G, C, T, T, A, G, A. So I went through there, and now we're going to add these in. And so again, remember, it's always going to grow 5' to 3'. So starting with this A right there, what would be complementary to the A? Well, it should be a T, but since it's RNA, we've got a U. G gives us a C. A gives us a U. A gives us a U. T does give us A, T, A, C, G, G, C. So this now is our strand of mRNA. And so there's the complementary base pairing that takes place during transcription. Okay, so just remember, it's the same as what we saw for DNA. The only difference is anytime that we should see a T at thymine, we replace it with a U for uracil. And then real briefly, we saw during translation, here's one where you really can't do it unless you've got this chart right here. Without this chart, it's going to be kind of difficult to do it unless you got a better memory than I do. But remember, we're going to take our mRNA, we're going to break it up into codons, which are 3 base pairs, and then we're going to read it from the chart. So if we had the codon CAU, the anticodon would be GUA, and that would be found on the tRNA. We really don't need to do that step, but you need to understand it. So let's find CAU on the chart. So let's look at our first nucleotide. We've got a C. The second nucleotide is an A, and then we've got U. So CAU is right here, and there is our amino acid. And we can do the same thing, make up any one you want. Let's go GUU. And so we can do the same thing. We first would be G, second U, third U, and we get valine. So now those three skills, if we put those all together, we can actually go from DNA to RNA to protein. So here's an example right here. We can see we've got double-stranded DNA at the top, right here. And we're going to transcribe, in this case, they're transcribing the bottom strand. So TAC becomes AUG, which happens to be methionine, the start codon. So each group of three gives us an amino acid. So this is what you'll have to be able to do in an example. If you're given a strand of DNA, to be able to transcribe it to mRNA, then translate it with the use of the chart into a polypeptide chain. And here's one more example from DNA to RNA to protein. So the whole central dogma laid out for you. So again, in this case, again, they are copying the bottom strand. So TGG becomes ACC, which translates to, I think it's threonine, an amino acid. So you can see it's broken into our triplets, the codons, that give us a specific amino acid and go through. So now let's actually try this together. And I've got the chart in the lower right for you to see if we can make this work. So what I've given you here is the DNA sense strand. So this is the one that we're going to read. Now realize there would be another strand here. If we looked at the top, it would be C, T, T. We could go through and fill in the rest, but we don't need to. So for now, all that I want you to look at is just, this is the strand of DNA. It is DNA that we're going to copy, and we're going to turn it into our mRNA. So G becomes C, A would become T, but it's RNA, so U, T becomes A becomes U, and G becomes C, A becomes U, G, A, U, G, A, U, T, excuse me. Let's go back one there. So T becomes, see, this is where you got to be careful. T becomes A becomes U, C, A should become T, but it's going to become U, C, G, A becomes U, T becomes A, G, A. So this now is our mRNA. And now we can go through, and I'll give you guys a second to look at this. If we go CUU, CUU, there's our first one. So that's going to be leucine. AUC, AUC, isoleucine. UUU, phenylalanine. GAA, GAA, glutamine. UGA, UGA, and that brings us to a stop codon. So in this case, we would read it, and this is where it would stop. The ribosomes would detach, and this would be our polypeptide. If you want a little bit of extra practice, you can pause the video and you could translate the last 4 codons if you wanted. But hopefully now this gives you an idea of how we can go from DNA to RNA to our polypeptide strand right there. So again, you're going to have a little bit of practice at this, work at it. Feel free to ask questions during class, and we'll get you some practice with that as well. So thanks again for your time.